In order for pre-mRNA to become a functional and mature mRNA that can travel outside of the nucleus, it must undergo a process called RNA splicing.
The pre-mRNA is the product of transcription and is composed of introns and exons. The introns don't code for any peptide sequence and must be removed. The exons contain the code for proteins and will be covalently joined together after introns and exons have been removed. The pre-mRNA also contains a 5' and 3' splice site on either side of what is known as the branch A site on the exon. These components are recognized by the spliceosome.
The spliceosomes, which is composed of 5 subunits called SNRPs, U1, U2, U4, U5, and U6. The spliceosomes catalyze reactions which allow for removal of introns.
U1 binds to the 5' GU splice site. U2 binds to the branched A site. U4, U6, U5 all form a trimer that attaches to the intron region.
The intron loops out and the exons are brought closer together. The 5' splice site is cut and the 5' end of the intron covalently attaches to the 2' OH group of the A on the branch site.
This forms a structure known as a lariat. The U1 and the U4 subunits are then released.
The 3' splice slice is cut and the exons covalently attach. U5, U6, and U2 stay attached to the lariat configuration until the intron becomes degraded and the SNRPs can splice other pre-mRNAs.
Although the introns don't code for anything, their purpose becomes advantageous in the process known as alternative splicing. This means variation can occur between the combinations of exons. Ultimately, the advantage is that 2 or more proteins can be derived from one single gene.
For example, take this pre-mRNA with exons from A through D and introns labeled 1 through 3.
If we focus our attention to exons A and B with intron 1 between them, and let's say the 3' AG splice site on the B exon is bad, the intron cannot be removed from this location. Therefore, exon B will be taken up by the spliceosomes with introns 1 and 2 as shown. The resulting mature mRNA will only be composed of exons A, C, and D. Thus, alternative splicing will allow for variation within the exon combinations.
The mature mRNA contains 3 components: the spliced exon sequence, a 5' methylguanosine cap, and a poly A tail. Let's first discuss the 5' cap. It is added to the pre-mRNA sequence by RNA polymerase after approximately 100 nucleotides have been synthesized by the RNA polymerase to the pre-mRNA chain.
The nucleotide at the 5' end of the pre-mRNA sequence has 3 phosphate groups.
Capping occurs in 3 steps. First, the RNA 5' triphosphatase leaves the 5' end of the pre-mRNA in preparation for addition of the cap.
The enzyme guanyl transferase removes one of the phosphates by hydrolysis and attaches the guanosine monophosphate, or GMP, to the 5' end of the pre-mRNA.
To complete this process, the enzyme methyltransferase attaches a methyl group to a nitrogen at the position 7 of the guanosine base.
The 5' cap is highly regulated and is vital for a stable, mature mRNA to undergo translation and is recognized by cap-binding proteins. The 5' cap also helps to protect the mRNA from exoribonucleases that could degrade it. Now let's focus our attention to the poly A tail.
The process of adding a poly tail is known as polyadenylation.
The poly A tail is located on the 3' end of the sequence. It is composed of a chain of adenosine nucleotides on the 3' end.
The polyadenylation signal sequence in eukaryotes is AAUAA. The endonuclease cleaves about 20 nucleotides downstream from this sequence.
This process occurs when the endonuclease cleaves the 3' end and poly-A polymerase adds adenine nucleotides to the 3' end of the mature mRNA. The maximum length for a poly-A tail is about 250 nucleotides.
Snip!
The poly-A tail, like the 5' cap, serves as a protection of the mRNA from exoribonucleases that may try to degrade it.
Excellent.